Okay, so this is week 3 in physiology. This week we will be looking at the muscle structure and muscle physiology. Looking at our body, 40% of it is made up of by skeletal muscle, with another 10 that make up smooth and cardiac muscle. When you look at some of the basic functions, first thing, again, we had discussed this in previous lectures, is that muscles, they attach to bones and they provide for movement. So it is the contraction of muscles of the bones that enables us to move different parts of our body. It allows us to move our entire body from one place to the other as well. Aside from that, skeletal muscles, they will also pull on soft tissues of the face, which allows us to have facial expressions. Aside from that, the movement that's caused by our respiratory muscles allows us to breathe. Another function, support. So muscles of the body wall, they support the internal organs. As these muscles lose their tone, the internal organs of the abdominal pelvic cavity may bulge outwards, as we see in some people, especially when they start to get older as they age. Protection. Skeletal muscles, particularly again of the body wall, they cushion the body's internal organs, within the abdominal cavity, from forces that may be applied by the exterior of the body. On very cold days, and when you're exercising, skeletal muscles will produce heat. Heat is a waste product of muscle metabolism, and under normal conditions, it helps maintain internal body temperature. So again, when we shivering is a mechanism of the muscular system that generates heat to warm up our body when it's overly cooled. Also, blood circulation. So cardiac muscle is what is pumping blood throughout our body. So the heart is the pump for the circulatory system. What we're able to see over here is that, again, we have different types of muscle. There's skeletal muscle, there is cardiac muscle, and there is smooth muscle. So these are specific types of muscles. So skeletal muscle, this creates movement in the body. Again, we have about 700 skeletal muscles that make up about, again, as we said earlier, roughly 40% of our mass, our body weight. And then remember what we said, the remaining 10 is smooth muscle and cardiac muscle. So cardiac muscle we only find in one area of the body, right? That is the heart, and it causes the heart to contract. And when the heart contracts, it pumps blood out of it. Smooth muscle will cause involuntary muscle contractions that we find lining not only in our intestines, but also within the blood vessels, for example. So these are just two areas that I've mentioned to you. You find them in blood vessels, you can find them in intestines as well. So again, the main thing to understand is that smooth muscle, involuntary, whereas skeletal muscles, they are voluntary. We have conscious control over them. Electrical excitability is a property of both neurons as well as muscle cells that allows them to respond to certain stimuli by producing action potentials. This means that they're able to generate electrical impulses that can travel along their membrane and transmit signals throughout the body. Now, contractility is the ability of muscles to contract forcefully when they are adequately stimulated. So when a muscle contracts, it generates tension, which can lead to movement if the muscle is attached to a joint, or if it's attached to skin. It will move to facial expressions. Right? Finally, the extensibility is the ability of muscles to stretch without being damaged. Okay? Okay, let's look at the chemical composition. So muscles are made of chemical components that work together to enable its function. One of the primary components is proteins, which makes up about 20% of the muscle. These proteins include contractile proteins, which are responsible for the muscle's ability to contract and generate force. In addition to that, enzymes, which are involved in various metabolic processes within the muscle. Another important component of muscle is ATP, adenosine triphosphate, as well as ADP, which is a byproduct, adenosine diphosphate. Okay? So ATP is the primary energy source for muscle contraction, and the breakdown of ATP into ADP releases energy that powers the muscle's movements. Myoglobin is another important component of muscle. This is a protein that stores oxygen within muscle cells. This allows muscle cells to have a readily available source of oxygen during periods of increased demand, such as when we're exercising. Finally, lactic acid. Lactic acid is a byproduct of muscle metabolism that can build up in the muscle during periods of intense activity. It's produced when the body breaks down glucose for energy in the absence of oxygen. So, while lactic acid can cause fatigue and muscle soreness, it is also a fuel for the body, and it can be converted back into glucose for energy. Skeletal muscles are made up of numerous fibers that range in size from 10 to 80 micrometers in diameter. These fibers are the basic units of skeletal muscle and are made up of smaller subunits. In most skeletal muscles, each fiber extends the entire length of the muscle. This means that the muscle is made up of a series of fibers that run parallel to each other, with each fiber spanning the entire length of the muscle. The fibers are organized into groups that are separated by connective tissue, which provides support and structure to the muscle. The number and arrangement of these fiber groups can vary depending on the muscle and its function. Okay, so the structure of skeletal muscle can be described as a hierarchy of increasingly smaller units. At the highest level is the whole muscle. And the whole muscle is an organ that's made up of multiple tissue types that include muscle fibers, connective tissue, and blood vessels. The muscle fibers are grouped together into fasciculi. Fasciculi are bundles of fibers that are surrounded by connective tissue. Each individual muscle fiber is a single cell that's made up of many myofibrils. Myofibrils are specialized structures within the cell. The myofibrils are made up of thick and thin filaments. Okay, so the myofibrils are made up of thick and thin filaments that slide past one another to generate muscle contraction. The thin filaments are made up of proteins called actin, while the thick filaments are made up of proteins called myosin. So we'll be looking at both of these in detail in this lecture. Both actin and myosin are key components in the process of muscle contraction. So when a muscle is stimulated to contract, the myosin heads interact with the actin filaments, pulling them closer together. This is what generates tension in the muscle fiber. And this process will continue throughout the muscle fiber, and when all the muscle fibers within the muscle are contracting together, the muscle as a whole generates force and produces movement. The sarcolemma is the outer membrane of a muscle fiber, and it's made up of two layers. The inner layer is a true cell membrane called the plasma membrane, which encloses the muscle fiber. The outer layer is made up of a thin layer of polysaccharide material that contains numerous thin collagen fibrils, which provide support as well as stability to the muscle fiber. At the end of each muscle fiber, the surface layer of the sarcolemma fuses with the tendon fiber. Tendon fibers are tough, flexible connective tissue that attach muscles to bones, allowing for movement and stability. These tendon fibers then collect into bundles to form the muscle tendons that insert into the bones. The sarcolemma and tendon fibers work together to allow muscles to generate force as well as movement. The sarcolemma encloses the muscle fibers and provides a barrier between the inside and outside of the cell, while the tendon fibers provide a strong attachment point to bones, allowing the muscles to generate movement and transmit force to the skeleton. Myofibrils are the fundamental building blocks of muscle fibers. They consist of two kinds of filaments, actin and myosin, which are responsible for muscle contraction. The myosin filaments are thick, while the actin filaments are thin. When these filaments interact, they create the necessary force for muscle contraction. Each muscle fiber contains hundreds to thousands of myofibrils. These myofibrils are made up of myosin and actin filaments, as we said. The myosin filaments consist of large polymerized protein molecules that interact with the actin filaments to cause muscle contraction. The actin filaments also consist of large polymerized protein molecules that have a specific structure and are responsible for interacting with the myosin filaments. What we see are this muscle here, and now let's look at but it will essentially if I because of these over another drawing that shows So in this representation, we're looking at microfilaments. We're looking at a small section of it, which we refer to as a sarcomere. And again, we'll talk about these in detail as we progress. But pretty much what they're showing you is that, look, we have these blue lines which represent thin filament. We have the red lines which represent the thick filaments. And it's the interaction with these thick and thin filaments that end up leading to muscle contraction. So again, we're looking at these areas called the sarcomeres. Let's look at the next slide, which shows another view of the same thing, perhaps a little bit more clarity, with better visualization. So looking over here, again, what are we looking at? This is a muscle fiber, and now we're taking a small section. So we're going from one Z disc to another Z disc. So this region from Z disc to Z disc is referred to as a sarcomere. So when we zoom in on this sarcomere, we see that it's made up of thin as well as thick filaments. Okay? So now, when you look at the area where there's only thick filament, okay, only thick filament that's present, no thin filament, that is referred to as the H zone. Okay, so in H zone, we only find thick filament. Okay. Now, the area where we have thick filament as well as thin filament would be the A band. Okay, so A band, we have both thick and thin filament. Now, the area where there's only thin filament and no thick filament is called the I band. Okay, So I band, only thin filament. H zone, only thick filament. A band has both thick as well as thin filament that's present. The mid part of the H zone is called the mid part of the H zone is called the M line. Okay, this is this part right over here, the M line. So there are three types of muscle proteins. First are the contractile proteins, and they generate force during muscle contraction. The next are the regulatory proteins, and these proteins help switch the muscle contraction process on and off. Then there are the structural proteins, and these are the proteins that keep the thick and the thin filaments of the myofibrils in proper alignment, which gives the myofibrils elasticity as well as extensibility. It also links the myofibrils to the sarcolemma and the extracellular matrix. All right, contractile proteins. As we said earlier, there's contractile proteins, and there's two types of contractile proteins, myosin and actin. So myosin is a contractile protein that makes up the thick filament, which is what we're looking at over here. So a myosin molecule consists of a tail and two heads. Okay? These heads binds to the myosin binding sites on the actin molecules of thin filaments during muscle contraction. Thin filaments, which isn't shown over here, which will be seen in the next slide, are contractile proteins. That's the main component of thin filaments. Okay, so actin is a contractile protein. That's the main component of thin filament, and each actin molecule has a binding site to which a myosin head of the thick filament will bind to during muscle contraction. So if you look over here, we have actin binding site on this myosin head, as well as ATP binding sites on this myosin head. We also see these light chain structures that are represented by the purple, and then you can also see the heavy chains over here that we find in the tails. Okay. Looking at regulatory proteins, and there's two types, tropomyosin and troponin. Tropomyosin is a regulatory protein that is one of the components of thin filaments. Now, in a relaxed skeletal muscle fiber, tropomyosin covers the myosin binding site on actin molecules, and this prevents the myosin from binding to the actin. Okay? It blocks the attachment site. Okay? Troponin is a regulatory protein that's a component of the thin filament as well. When calcium ions bind to troponin, troponin undergoes a conformational change that moves the tropomyosin away from the myosin binding sites on the actin molecules. And this will allow muscle contraction to start, because now you have the myosin sites that are exposed, which will allow for the myosin to bind to actin. Now let's talk about these structural proteins. First one is titin. Titin is a structural protein that connects a Z disc to the M line of the sarcomere, and this is for helping it to stabilize the position of the thick filaments. Because it can stretch and then spring back together without any damage, titin accounts for much of the elasticity as well as the extensibility of microfibrils. Next, we talk about alpha-actin. This is a structural protein of the Z disc that attaches to actin molecules of thin filaments and to titin molecules. Myosin. Myosin, again, this is a structural protein that forms the M line of the sarcomere. Okay? It binds to titin molecules and connects the adjacent thick filaments to one another. Next one to talk about is nebulin. This is also a structural protein that wraps around the entire length of each thin filament. It's going to help anchor the thin filament to the Z discs, and it regulates the length of the thin filament during development. Last, dystrophin. Dystrophin is the structural protein that links the thin filament of the sarcomere to the integral membrane proteins in the sarcolemma, which are attached in turn to proteins in the connective tissue matrix that surrounds muscle fibers. There's also thought that dystrophin helps reinforce the sarcolemma and helps transmit tension that's generated by sarcomeres to the tendons. Right. The myosin and actin filaments in the myofibrils partially interdigitate, creating an alternating light and dark bands that we saw on the previous slides. The light bands are also known as I bands, and I bands, as we said, contain only actin filaments. I bands are also isotropic to polarized light. The dark bands, which we said, are the A bands. They contain the myosin filaments and the overlapping ends of the actin filaments. Now, the A bands are said to be anisotropic to polarized light, and this is because of the alignment of the myosin filaments. So the actin filaments in myofibrils are attached to the Z disc, which are made up of different filamentous proteins than the actin and myosin filaments. The Z disc passes crosswise across the myofibrils and also crosswise from myofibril to myofibril, attaching the myofibrils to one another all the way across the muscle fiber. The end of the actin filaments are attached to the Z discs, and from there they extend in both directions to interdigitate with the myosin filaments. This arrangement is what allows for the actin and myosin filaments to slide past each other during muscle contraction. The portion of the myofibril between two successive Z discs are what we called a sarcomere. When the muscle fiber contracts, the length of the sarcomere is about 2 micrometers. Now, at this length, the actin filaments completely overlap the myosin filaments, and the tips of the actin filaments are just beginning to overlap one another. This length produces the greatest force of contraction because the maximal number of actin-myosin cross bridges are able to form. And this allows for the most effective interaction between the filaments as well. So, the sarcomere length is an important determination of the muscle force production. In this slide, again, what we're looking at is what do we have here? So look, here's a sarcomere going from one Z disc to another Z disc. You can see the M line over here, and we see the thick filaments, which are represented in purple, and the thin filaments, which are in red. So again, at each one of these things, you can see, look, when you're looking at the I band, what do we see in the I band? In I band, we see only the thin filaments. Then when you start looking at the H zone over here, H zone, we see only the thick filaments. All right? Again, in the M line, we see thick filaments that are linked, as you can see over here, with some of these accessory proteins that we have. And then when you start looking at the outer edge of this A band over here, so here we go, right around here, what do we see? This area. We see both thick and thin filaments that are overlapping. All right, so the side-by-side relationship between the myosin and actin filaments in muscle fibers is critical for muscle contraction. However, maintaining this relationship is not easy, it's difficult, and this is due to the dynamic nature of the filaments during contraction and relaxation. So, to address this challenge, titin comes to help out. Okay, so titin is the protein that we talked about earlier. It's a large filamentous molecule. It acts as a framework holding the myosin and actin filaments in place. It's also thought to be the largest protein in the body. So by anchoring the myosin and actin filaments in place, titin helps to make sure that the contractile machinery of the sarcomere will work properly. And this allows for the efficient muscle contraction and force production to take place. Okay, the intracellular fluid that fills the space between myofibrils in muscle fibers is called sarcoplasm. Okay? Sarcoplasm contains a high amount of potassium, magnesium, and phosphates, as well as numerous protein enzymes. Now, one of the most important components of the sarcoplasm is the mitochondria. Okay? And mitochondria are the organelles that supply the contracting microfibrils with large amounts of energy that they need. And this energy comes in the form of adenosine triphosphate, or ATP. And ATP is necessary for muscle contraction to take place. And the higher concentration of mitochondria in the sarcoplasm ensures that there is a constant supply of ATP that's available during periods of intense activity. All right, the sarcoplasmic reticulum is a specialized endoplasmic reticulum that's found in skeletal muscle fibers. It's located in the sarcoplasm surrounding the myofibrils and has a unique organization that's crucial in controlling muscle contraction. The SR, sarcoplasmic reticulum, functions as a calcium store and release system. When an action potential triggers the release of calcium ions from the sarcoplasmic reticulum, it's going to cause the myosin and actin filaments to interact with one another. And this is what results in muscle contraction. Now, the organization of the sarcoplasmic reticulum is particularly important in controlling muscle contraction. Rapidly contracting muscle fibers have especially extensive sarcoplasmic reticula to ensure a rapid and efficient release of calcium ions. So over here, you can see, what do we have here? These are, well, right over here are the lateral sacs. We have the myofibrils over here. And again, you can see I bands, you can see the A bands over here, you can see the M line here. And then these are the transverse tubules, or T tubules, sometimes as they're called. And over here we have the sarcoplasmic reticulum, the SRs, right over there. Okay. So when you look over here, what we see is one Z disc, another Z disc. So the area between the Z disc is what we refer to as a sarcomere. And then within the sarcomere, we will see our thick as well as thin filaments. Again, in the sarcolemma, here's the sarcolemma, okay, and this is the sarcoplasm. So in the sarcoplasm, what do we see? We see a lot of mitochondria that's present, that we said it's crucial, right, to producing energy to sustain contraction, because muscle cells require lots of ATP, lots of energy is needed. Also, you can see the sarcoplasmic reticulum that's found over here. The myosin molecule is made up of 2 heavy chains and 4 light chains, forming a double helix structure called the tail. The myosin head is formed by folding one end of each heavy chain into a globular polypeptide structure. The myosin molecules play crucial roles in muscle contraction by binding the actin filaments and using ATP hydrolysis to generate the necessary energy for muscle movement to take place. The myosin filaments made up more than 200 myosin molecules, with their tails bundling together to form the body of the filament. The head of these myosin molecules extend outwards to the sides of the body, and this is what forms the flexible cross bridges. Now, these cross bridges, they play a crucial role in muscle contraction by binding the actin filaments, and this is what forms this ratcheting mechanism that generates force as well as movement. The flexibility of the cross bridges is what allows them to undergo a conformational change during muscle contraction, and this results in the sliding of the actin and myosin filaments past one another. The myosin filaments contain the cross bridges, which again are small projections from the side of the filaments. The interaction between these cross bridges and the actin filament is what causes the contraction of the muscles. So myosin filaments again, what did we say? These are thick filaments in muscle fibers. They contain the small projections that we call the cross bridges. The cross bridges, they stick from the sides of the myosin filament and play the role in the contraction. So it's during this contraction, the cross bridge of the myosin filaments interact with the thin filament of the actin, which are found arranged in a double helix with the tropomyosin and troponin molecules. So the myosin heads attach to the actin filaments, forming a cross bridge, and then they swivel, pulling the actin filaments toward the center of the sarcomere. And this is what results in the contraction of the muscle fiber. So if you look over here, these are the myosin heads, and these are the tails over here. Okay, so these heads, when they attach to the thin filaments, and again in the presence of ATP, they will swivel, and it creates this retching-like action, this movement, and this is what ends up pulling the actin molecules over one another. It causes an overlap, and this is essentially contraction, what contraction is. Alright, so the myosin heads has an important property that is necessary for muscle contraction, and that is its ATPase activity. This means that the myosin head can act as an enzyme and break down ATP molecules, releasing that energy that's stored in the high energy phosphate bonds. So the energy is then used to fuel the contraction process. So therefore, this ATPase activity of the myosin head plays a very, very critical role in the sliding filament theory of muscle contraction. So over here, we can see that the myosin molecule has two binding sites. Right? One is an actin binding site, and then the other is the ATPase site, right over here. The actin filament is made up of a double-stranded F-actin molecule. Each strand of the helix is made up of polymerized G-actin molecules. The F-actin backbone is a highly dynamic structure and has the ability to rapidly assemble and disassemble in response to various signals. The G-actin monomers can bind to ATP and hydrolyze it to ADP, which provides energy for actin polymerization. The actin filaments they're essential components of the cytoskeleton as well. If you remember back to the first couple of lectures, we saw this is showing. Actin, again, it's crucial for the cytoskeleton as well as other cellular processes, including cell movement, including cytokinesis, and also they play a role in intracellular transport. So this is a very, very, very important protein. The interaction of actin filaments with the myosin cross bridges plays a critical role in muscle contraction. So we find them inserted firmly into the Z discs, which are these specialized structures that we find at each end of the sarcomere. And the Z discs are what provides an anchoring point for the actin filaments, which allow them to stay in place during muscle contraction. The thin filaments extend in both directions from the Z disc, with their ends lying in the space between the myosin molecules. The myosin cross bridges we find located on the surface of the myosin filaments attached to the actin filaments at these points. And this interaction between the myosin cross bridges and the actin filaments causes the filaments to slide past one another, and this is what results in muscle contraction. The actin filament contains the protein tropomyosin, which is wrapped around the sides of the double F-actin helix. In the resting state, the tropomyosin molecule lies on top of the active sites of the actin strand, okay, which you can see over here. This is what prevents the attraction between the actin and myosin filaments and prevents the contraction. Why? Because it's blocking the binding site for the myosin head. Now, troponin is a protein complex that's made up of 3 subunits: troponin I, troponin T, and troponin C. Troponin I has a high affinity for actin. Troponin T binds to tropomyosin, and troponin C binds to calcium ions. So the complex is responsible for attaching tropomyosin to actin. In a resting state, the tropomyosin molecule lies over the active sites of the actin, and this prevents the attraction between actin and myosin filaments to initiate contraction. When calcium ions bind to troponin C, the troponin complex undergoes a conformational change, and this causes the tropomyosin to move away from the active site of the actin filament, which is going to allow the cross bridge of the myosin to bind with actin and initiate the contraction process. So the troponin-tropomyosin complex plays a crucial role in regulating muscle contraction. In a relaxed muscle, the active sites on the actin filaments are physically covered by the troponin-tropomyosin complex, which we looked at in the previous slides. And again, we said that this is what prevents the attachment of the myosin cross bridge heads, and it inhibits contraction. We also said that when calcium ions bind to troponin C, it induces a conformational change in the troponin complex, which uncovers the active sites on the actin filaments. So now that these sites are exposed, it's going to allow them to bind to the myosin cross bridge heads, and this is going to initiate the contraction process. So you can see over here again, what do we have? We have the tail of the myosin, and we have the head of the myosin, right? So you got the heads, which are the light chains, and then the tails, which are the heavy chains. Then, so this is all, this is part of the thick filament, right, over here. So now we look over here at the thin filament, and over here on the thin filament we see the actin molecules, and you can see the troponin, and you can see tropomyosin that are covering the binding sites of actin on the actin molecule. Right, going over the action potential. So firstly, the resting membrane potential of skeletal fibers is between minus 80 to minus 90 millivolts, okay, which is similar to myelinated nerve fibers. Remember, in the previous lectures we said it's about minus 70 or so. Now, secondly, the duration of the action potential in skeletal muscles is about 1 to 5 milliseconds, which is approximately 5 times longer than in large myelinated nerves. Third thing that we have over here is that the velocity of conduction in skeletal muscle is 3 to 5 meters per second, which is about 1/13 of the velocity of conduction in large myelinated nerve fibers that excite skeletal muscles. So the initiation and execution of muscle contraction occurs in sequential steps. First, an action potential travels along a motor nerve to its endings on muscle fibers. Then the neurotransmitter substance acetylcholine is secreted by the nerve ending, which acts on a local area of the muscle fibrous membrane. This opens multiple acetylcholine-gated cation channels through protein molecules floating in the membrane. The opening of these channels allows large quantities of sodium ions to diffuse to the interior of the muscle fiber membrane, and this causes a local depolarization. This in turn leads to the opening of the voltage-gated sodium channels, which initiates an action potential in the membrane. The action potential travels along the muscle fiber membrane, causing depolarization of the muscle membrane. Now, this depolarization causes the sarcoplasmic reticulum to release the stored calcium ions. The calcium ions initiate attractive forces between the actin and myofilaments, cause them to slide alongside one another to produce muscle contraction. The calcium ions, they're pumped back into the sarcoplasmic reticulum by a membrane pump, and the contraction process ends. All right. The sliding filament mechanism of muscle contraction occurs when the actin filaments, which extend from the two successive Z discs, overlap one another maximally in a contracted state. That's what we're looking at over here, right? What do we have? You can see that these thin filaments, they're overlapping with one another over here, okay? Now, in the relaxed state over here, these filaments, they barely overlap. So the Z discs have been pulled up to the ends of the myosin filaments due to the inward pulling of the actin filaments. And this mechanism causes muscle contraction. So what causes the actin filaments to slide inward among the myosin filaments? The sliding filament mechanism of muscle contraction is caused by the interaction of cross bridges from the myosin filaments with the actin filaments. Under resting conditions, these forces are inactive. When an action potential travels along the muscle fiber, this causes the sarcoplasmic reticulum to release calcium ions, which activate the forces between the myosin and actin filaments, and contraction begins. ATP molecule is degraded to ADP, adenosine diphosphate, to liberate energy for the contractile process to move forward. So over here, what we're looking at is this. Look, this is during a relaxed muscle. Notice what we see. The actin filaments, they're far apart, right? The myosin heads, they're not attached at this point, so there's no contraction. Now, in a partially contracted state, look what's happening over here. Look at the H zone. Look at the distance, how wide the H zone is. And now look over here. The H zone is much more narrow. So now the myosin heads are starting to pull on these actin molecules. And when you look at a maximally contracted state of muscle, look what happens. The actin molecules, they overlap with one another. Okay, this is complete maximum contraction. All right, the contraction cycle of muscle fiber involves 4 major steps. First, we have ATP hydrolysis, and this provides the energy that's required for muscle contraction. Second, we have attachment of myosin to actin that needs to occur. This is what's going to allow the myosin cross bridges to form. Third, we have the power stroke. The power stroke takes place as the myosin cross bridge pull the actin filaments towards the center of the sarcomere. Finally, we have detachment of myosin from the actin that occurs. And this allows the cycle to repeat and the muscle to contract further. All right, so let's go over this the contraction cycle. So we'll start off with number 1. So ATP hydrolysis. This is what's taking place in step 1. So over here, what's happening is, again, as I said, ATP hydrolysis. Now remember what we said earlier. The myosin head, it includes the ATPase site, right? So this is the ATP binding site that will hydrolyze ATP into ADP, adenosine triphosphate, into adenosine diphosphate. And the energy that's generated from this hydrolysis reaction is stored in the myosin head for later use during the contraction cycle. So at this point, the myosin head is it's said to be in an energized state, okay, and it assumes this 90-degree angle relative to the thick filaments that you see over here, thick and thin filaments. So when this happens, it has the proper orientation to bind to an actin molecule. Also notice at this point that the product of ATP hydrolysis, again ADP and P, they're still attached to the myosin head. Right, so that's number one. And the second step is the attachment of myosin to actin. So this energized myosin head attaches to myosin binding sites on the actin, and it releases this previously hydrolyzed phosphate group. When a myosin head attaches to the actin during the contraction cycle, the myosin head is referred to as a cross bridge. So now we have cross bridge over here, right? So now, even though a single myosin molecule has a double head, only one head binds to actin at a time. Now let's move on to the next step over here. So this is the power stroke. So after a cross bridge forms, the myosin head, it pivots. Remember over here we said it's at 90 degree. Now look what happened, 45 degree. Okay, so the myosin head, it pivots. It goes from that 90 degree angle to a 45 degree angle relative to that thick and thin filaments. So as the myosin head changes to its new position, it pulls, it pulls the thin filaments past the thick filaments towards the center of the sarcomere. And this is generating tension. It's generating force. This is what we call the power stroke. Okay? And the energy that's required for this power stroke we got from over here in step one. In the last step, the detachment of myosin from the actin. So over here, at the end of this power stroke, notice that the cross bridge, it remains attached. It's firmly attached to the actin. And this will be the case until it binds another molecule of ATP. So as the ATP binds to the ATP binding site on the myosin head detaches from the actin. Okay, so the neuromuscular junction is the synapse between a somatic motor neuron and a skeletal muscle fiber. At this junction, the end of the motor neuron forms the motor end plate on the muscle fiber membrane. When the motor neuron is activated, it releases the neurotransmitter acetylcholine. Acetylcholine binds to receptors on the muscle fiber membrane, causing depolarization of the membrane and generating an end plate potential. So if the end plate potential reaches a threshold, an action potential is generated at the muscle membrane. That will travel along the muscle fiber and initiate muscle contraction. The motor end plate, this is a region of the muscle fiber membrane that's opposite the synaptic end bulb. This is a very brief overview of what we have learned in the second week. They're showing you over here the resting membrane potential. And in this case, again, they're stating it's minus 90 millivolts. Over here, we have depolarization that's taking place. Of course, we reach depolarization after meeting a threshold. Okay, so, and then the peak is roughly at positive 30 millivolts. And then after that point, we have polarization that takes place, and then that repolarization returns back to a resting membrane potential. Again, this is a very simplified waveform that they're showing you. We looked at a much more detailed and much more accurate graphics in our previous lecture. So if you have forgotten, be sure you go back and you review that. This is extremely basic, and it's just telling you what's happening in that when we have depolarization taking place, we have sodium channels that are open, and then during the repolarization process, we have the potassium channels that are open, and the sodium channels, they will close. Now over here, excitation, contraction, coupling. Okay, this is showing a relaxed state. So, all right, what's happening over here? In a relaxed state, very simplistically, what they're showing you is this. This is a muscle cell, okay, or inside here. We have, this is a sarcolemma over here. Remember, from one Z disc to the other Z disc is a sarcolemma. So now you have the thick and the thin filaments over here. So this is the membrane, right? This is a sarcomere, This is the sarcomere, then this is the sarcolemma, or the membrane of the muscle cell. All right. So if this sarcolemma, if there is an action potential that travels along it, we will get contraction that takes place, right? So in this picture, it's showing relaxed. So what's happening in a relaxed state? So again, this is the sarcolemma over here. And remember, inside the muscle cells, there are sarcoplasmic reticulum, the SR, sarcoplasmic reticulum. And the sarcoplasmic reticulum, they store calcium ions, all right? And these calcium ions, they have gatekeepers that allow for their proper release, their uptake, which are done by these guys over here, okay? These are these calcium release channels. And we'll talk a little bit more about them, but essentially, very simplistically, they have these calcium release channels, they have two parts that they're made up of, two units. They have this dihydropyridine receptors that they are made up of, and then they also consist of a foot process that they are made up of. Okay. So the ryanodine receptors, and then we have the dihydropyridine receptors. Right? These are the two components of this calcium release channel. All right. So in a relaxed state, these are blocking the calcium from escaping the sarcoplasmic reticulum. Okay, that's what's happening in a relaxed state. Very simplistically, this is what you're seeing. Because remember what we said, if you want muscle contraction to occur, what needs to happen? The myosin heads need to attach themselves to the thin filaments, right? And we said that can only happen when we have a conformation of the tropomyosin that's blocking the binding sites on the actin. Okay, so here's the myosin binding sites on the actin molecule. Here's tropomyosin that's sitting on top of it. That's preventing the myosin heads from attaching onto it. Then we'll also have these troponin molecules over here, which are kind of helping hold this tropomyosin in place. So when calcium is released, calcium will bind to this troponin, and it will push this tropomyosin off the binding sites for actin, where the thick filaments can then come and attach themselves onto. Okay, that's simplistically what's happening. What's going to happen? For the myosin head to attach onto the thin filament. So, again, in order for that to happen, we need calcium. Right now, the calcium is closing, and this is when the muscle is relaxed, in a relaxed state. Now, contraction. Okay? Now, contraction. What happens? We have an action potential that comes. As the action potential comes, what's going to end up happening? It's going to cause a conformation change in these gatekeepers, right? In the calcium release channels. And what happens? You see these over here, the dihydropyrimidine receptors. You see that they kind of move off, and now you got calcium that starts to leak out. Okay. So as the calcium starts to leak out, what's going to happen? They're going to go and they're going to attach onto the troponin. As they attach onto the troponin is then going to push the tropomyosin off the anti-actin binding sites. Now you can see these black things are exposed. Now the myosin heads can come and attach to these myosin binding sites on the actin molecule. Okay, so that's essentially it. This is what's happening. All right, so if we were to summarize this process, let's look at all the steps that are involved. First things first, we'll start off at the neuromuscular junction, right? Nerve and muscle, neuromuscular junction. So an action potential is propagating down the axon. It comes to the axon terminal. Okay. Over here we have synaptic vesicles. Inside the synaptic vesicles, there's neurotransmitters that are stored. Okay. What are these neurotransmitters? They are acetylcholine. So acetylcholine will be liberated from the synaptic vesicles, and they will fall. this is, remember what we said, this is a muscle membrane over here. They will fall where? Onto the motor end plate. So, and on this motor end plate, there are receptors. Okay, there are ligand-gated channels, and these ligand-gated channels, they are the ligand that they're sensitive to or responsive to are acetylcholine. So acetylcholine will fall off. It attaches onto these ligand-gated receptors, and which will cause a change in conformation. Right? It causes these channels to open up. As they open up, now sodium can enter. When sodium enters, we have depolarization that takes place. Okay, boom. So as this muscle membrane, it depolarizes, right? The sarcolemma depolarizes. What's going to happen? It's going to go to the sarcoplasmic reticulum and open up these calcium storage channels, right? It causes a change in the conformation of these machines over here, which are blocking the holding in the storage of calcium inside the sarcoplasmic reticulum. So when these things open, now we get calcium that starts to leak. As the calcium leaks, it's going to come and calcium is going to bind to troponin. Now notice over here, what are we looking at over here? This is a sarcomere, right? We're going from one Z-disc to the next, and this is in a relaxed state still. Why? Because there hasn't been calcium that's reached here. So once this happens, we come over here. Now calcium is able to bind to the troponin and cause a conformational change. Now the actin binding sites are exposed. Notice over here, again, over here, when there is no action potential in the relaxed state, these actin binding sites are blocked, okay, by the tropomyosin. So when the calcium is exposed and it binds to the troponin, now the tropomyosin gets moved by the troponin to expose these myosin binding sites. Okay, now we can have the myosin heads attach. So a handful of slides ago, when we went through those 4 steps, that's what's going to take place at this point. Okay, once the myosin binding sites on the actin molecule is exposed. Okay, now we will have the myosin head that's going to bind to actin, and it will undergo a power stroke. Okay, remember we need ATP for this to happen over here also. So once we end up having that contraction, next thing, what's going to happen after that? Okay, after you have the contraction that takes place, we have the next step over here in number 7, where the calcium ATPase pumps end up sucking up all of this calcium back inside the sarcoplasmic reticulum. So the calcium gets recycled, gets used up. And what is that? Calcium ATPase pumps. They will pump back the calcium back into the sarcoplasmic reticulum. Okay? And then what happens after that? Now the calcium is no longer there. The tropomyosin will slide back onto the actin binding sites to block the myosin heads from attaching. And the muscle relaxes. Okay? And this will repeat until the next action potential arises. All right. So that summarizes this entire concept of all the steps that we had learned. Be sure you review this a handful of times to understand this. All right, so that's it for this lecture. Thank you for watching this, and we'll see you again next time.